Hello, everyone. I am Alison Robertson, and I'm a professor and extension field crops pathologist here at Iowa State University. And in this webinar, I am going to be talking with my colleague, Tamra Jackson-Ziems, who's also a professor and extension plant pathologist at the University of Nebraska. And Tamra and I have been working on crown rot of corn um, in tandem. And so this webinar just gives you an update on what we've been finding. And the title of it is called Crown Rot, There's a Party Going On. And I think that'll become evident as we go through the presentation. So thanks all for joining us, and we hope we can let you learn a little bit more about crown rot. So I'm going to pass this to Tamra, I hope. There we go.
Okay. Well, crown rot is a disease that has been occurring in several states around the Corn Belt. And the symptoms that people recognize as crown rot may not become evident to most people until toward the end of the season as we approach senescence. And often above ground, the first thing we might notice is that some plants may die early. But leading up to that, for a few days, you might see some really odd discoloration. We call it ghosting, right? It's an off, ugly, greenish gray color that's been very common where the tops of the plant die. Well, these plants can be single plants or clusters of them, and the cause of that is a little bit unclear unless you dig up the root ball and then split that root ball to look at the crown. And as you can see in the pictures, you've got on the left, there's a photo of what ghosting looks like out in the field. And then you've got a photo of a rotted crown that's been split open. And that's a really severe case. It can be varying levels of that compared to what a healthy crown might look like, where it's still very white and clean on the inside. We also often see root rot associated with plants that have a lot of crown rot as well.
Thanks, Tamra. So one of the things that Tamra and I started by doing was trying to identify what the organism was that was causing crown rot. So what we needed to do for that is we needed to collect plants that were showing symptoms of crown rot. And then what we do is we take pieces of tissue from the edge of that symptomatic tissue, and then also from the healthy tissue, and we put those tiny pieces on tissue onto selective media, which is just basically Petri dishes that are filled with a jelly-like substance. And then if there's any causal organism or any pathogen, or just even if there's any organism associated with that piece of tissue, it'll grow out onto these plates. And so a lot of times we can tell just by looking at the plate what that organism might be. But to be really sure what that organism is, we need to sequence and identify that organism. So there's a couple of differences between my lab and Tamra's lab that I want to bring up here because we've had slightly different results, and that's probably because we were using slightly different media in our labs. So in Tamra's lab, the jelly that she was using was a media called water agar. And in my lab, the jelly that I was using was a half-strength potato dextrose agar media. And so the differences between those media are going to encourage different organisms to grow, or they might favor different organisms over other organisms. And then those organisms can also look quite different on one media compared to another media. So a lot of our initial work that were by our graduate students were collecting these symptomatic plants and then doing these isolations from the plants and sequencing and identifying the organisms that we found associated with that. Now, traditionally, crown rot has been called Fusarium crown rot because you always isolate fusariums from it. But in some work that we did back in 2022, yes, you can see that we isolated some fusarium species there, but we also isolated a lot of different genera and species. And so the question now becomes, which of these organisms is causing crown rot? And the way that I like to think about it is that crown rot is a party, right? And hence the title of this presentation. There's a party going on there. Every time we do these isolations, we isolate a lot of different organisms from them. And so the questions that we have are, whose party is this? Who was invited to the party? Whose gate crashed the party? And who is that in the golden mask? Because we don't recognize that person. We don't know where they came from. We don't know who they're associated with. And so these are questions that we're dealing with crown rot. Crown rot is obviously a complex of different organisms, but there's got to be one or a couple of organisms who start the whole crown rot infection process. So that's their party. Then we have other organisms that are invited, they come into the party, but then we have organisms that just gate crash the party, right? Those will probably be something like saprophytes that are there because they're feeding on the dead tissue. And then occasionally we find organisms that we have no idea what they are or what role they're playing in this crown rot. So as I mentioned, in Tamra's lab, they were using a slightly different media to what we were using in our lab. And so, Tamra, why don't you describe to our listeners what you found in your lab?
Well, sounds good. I think the metaphor is really good that this is a party, and we saw a different party. And just like you mentioned earlier, where fusarium species have been blamed in some studies historically, we did get a lot of fusarium species. In fact, we had 8 different fusarium species from our earliest efforts back in 2022. And so this was these were isolations made from crown rot symptomatic plants collected at R5 at the end of the season. And it's really, I don't think, a surprise for any of us working in pathology that we would find a lot of fusarium species because they are nearly ubiquitous almost everywhere. And many of the ones we are finding are important pathogens of not only corn but other plant species. Some of them are also groups. So Fusarium graminearum, for instance, was the most common fusarium species that we found from the beginning here, and you can see that 27% of those 1,200 plus isolates that we collected were actually Fusarium graminearum or in that complex. And there's a few species within that group that are common. And so that was interesting to see. Some of these other species may or may not be that important pathogens. Some of them are common colonizers of roots and crowns. And it's important for us to figure out, like you said, whose party is this? Who is to blame for symptom development? And are there other players that might be present or might help promote either infection or symptom development?
So if we go back to my lab, one of the things that we were interested in is that in our samples that we collected in 2022, we isolated this one fungus, Phytocytroma ambiguum, almost as frequently as we isolated Fusarium graminearum, which you'll remember was the most prevalent species that Tamra's lab isolated. And so this was of interest to us. And when we started to look into Phytocytroma ambiguum, which you can hear is quite a mouthful. And so in my lab, we have nicknamed it PAM because that's way easier. So I think from now on, I will just continue with PAM. But when we looked into reports of PAM from around the world, we found that PAM had been reported as causing crown rot in Australia, in South Africa. South Africa's probably done the most work on this organism. Europe, and then also Brazil. And so that kind of piqued our interest, and we wondered whether Pam could be playing, could maybe be the host of this crown rot party that was going on. So in 2023 and 2024, we collected crown rot symptomatic plants from across the Midwest. And this was in collaboration with Bayer. And the survey that we did with Bayer was also done in Tamra's lab. So we had samples coming to Iowa State and samples going to Nebraska. In my lab, we were most interested in which of those samples contained PAM. And so in 2023, of the 505 samples that we collected, the samples of tissue from those infected crowns, almost 50 of those had contained PAM, and about 35 of those were Fusarium species. In 2024, we received far less samples. The reason for that being permitting issues. But you can see that in 2024, we collected a lot more Fusarium species than we did PAM species. But we are collecting PAM from these crown rot symptomatic plants. And so that's been our goal, is to try and understand what role PAM is playing.
So the next step after we identify some of the microorganisms and especially pathogens that are being recovered from these symptomatic or sick plants is to try to identify who's there and then try to reproduce the symptoms, the crown rot symptoms that we're observing in the field. This process is a scientific process that we call Koch's postulates, and it's part of the process that we need to conduct and complete to prove which organism or group of organisms is causing disease. And there's a lot of ways about going about this. And in both Alison's lab and ours in Nebraska, we have students that have been working on this and taking these organisms, both PAM and some of the Fusarium species from our own collections, and inoculating them onto healthy plants in an effort to reproduce those crown rot symptoms. We do that in a lot of different ways, in liquid slurries of spores, suspensions of spores, so we do that in a lot of ways, including with spores, and we're inoculating with slurries or suspensions of mycelia. We're also inoculating toothpicks and using them to jab into the crown. We're also using colonized agar discs and other substrates like millet and sorghum, and doing that in inoculum layer in greenhouse pots like you see in the photos here. And even though we're both doing that work and have a lot of experience doing that and a lot of replications, we have continued to fail to duplicate those exact crown rot symptoms, except for when we directly stab the crown of the plants with those infested toothpicks. So we're trying a lot of different techniques, trying to inoculate these plants so we can reproduce those crown rot symptoms. And an example of what we're shooting for is to get crown rot symptoms like you see in the rotted photo there on the right-hand side versus the healthy plant on the far right side. Unfortunately, we've had a lot of failure in trying to reproduce these crown rot symptoms using almost all of those techniques. The one that has been the most successful is the toothpick technique, where we're literally infesting sterile toothpicks with the different pathogens and stabbing it directly into the crown of the plant. This was a surefire method, I guess, but it's not one that really is representative of processes that are going on in the field. And so, since that's the only one that's been working to duplicate those symptoms, like you see in the photo, you can see the toothpick hanging out the bottom of that plant and what the results look like when those plants are taken down and split. You can see where the crown rot symptoms are. And so, we don't feel like we've really got good conclusions and answers yet on which pathogen or pathogens are to blame.
Thanks, Tamra. Yeah, that's probably been some of the most frustrating part about this work, not only for us, but for our students as well. Taking these methods that have been published. I know the methods that we've used for PAM have been published, and yet we just can't replicate those results in our greenhouse. And so it makes it very hard for us to be able to say that pathogen X is it's their party, right? When we're not able to actually show it in the greenhouse. So the other thing about the toothpick method, right? You mentioned that we're just not happy about it, and um, it brings up a lot of questions as well. These different methods that we've been doing. Um, when does infection occur? So I know that your lab and my lab, we've been inoculating seeds, we've been inoculating um, young seedlings, we've been inoculating older plants, and yet we cannot get symptoms. How does the infection occur? We're not sure how that's happening. I know that your lab's been working on looking at nematode damage, or at least nematodes in the samples that you've been receiving, to see if that might be facilitating infection. We've done a little bit of work damaging the roots, like we might expect a corn rootworm larvae to damage those roots to see whether that might facilitate infection, but we just haven't been successful in those areas. And then the other thing is plant stress. So I know when we started working on crown rot, we thought that cold, wet conditions at planting facilitated the infection, and then stressful periods towards the end of the growing season kind of let the crown rot develop, and then we got those symptoms in the field. And yeah, sometimes I was thinking about that, because we're like, sometimes I think we're thinking of crown rot like stalk rot. And is crown rot a version of stalk rot? Does it develop like stalk rot? Are we barking up the wrong tree thinking about stalk rot? I don't know. Those are just some questions that I've been thinking of. I don't know if you have any thoughts along that line.
The same. We have a lot of questions and I guess concerns about, what are we missing? And is it possibly that two or more pathogens are working together, or can it be different combinations of pathogens that can cause the same symptoms that we continue to see? And so there's still a lot of unanswered questions.
Yeah, and absolutely. I mean, it's when you have so many pathogens, it's also hard to decide who to put together, right? I mean, hopefully we could put some together and get those symptoms to develop, but who do we put together? So, yeah. So we have been doing some small plot trials to investigate the effect of in-furrow and foliar-applied fungicides on crown rot development. And these kind of arose because I'd had some conversations with an independent crop consultant who works in the Corn Belt, and he felt like he was getting good control of crown rot with some in-furrow fungicides. And based on what we know about the effect of foliar fungicides on stalk rot, that they can help with stalk rot. Sometimes we wanted to do some small plot trials just to try and investigate what was going on there. And so in 2023 and 2024, we did these field trials down at Iowa State research farms. So in McNay and Southeast Research Farm down there in the South, we did the trials there in both the years. And then in 2023, we did the trial at Huxley. In 2024, we did the trial in Boone County on the ag engineering and agronomy farm. The reason why the ag engineering and agronomy farm was chosen was because we went into a field where we knew that we had had severe crown rot a couple of years previously. And so we absolutely knew that that field had a history of crown rot. The other thing that we did with these trials is we planted a crown rot susceptible hybrid, and that was treated with Acceleron, the standard seed treatment that Bayer uses. We also planted these trials early in both years. We planted kind of the middle of April, just before we got a cold, wet period come through. And then in McNay in 2024, we had two planting dates. We planted early and then we planted approximately a month later, just to try and see if there was an effective planting date. And you can see the treatments that we had there. Obviously, we have a check where we don't do anything, right? And then we had fungicide applied at V5, V6, and then at tasseling and silking. In 2023, we included a double application of fungicide. In 2023, we had headline in furrow. And in 2024, we used propulse in furrow. And what we did with these field trials is, excuse me, we went out several times during the growing season and we collected plants, split those plants open, and then did isolations from those plants to see what was growing in those crowns. And unfortunately, we didn't have any crown rot develop at any of those 3 sites, 4 sites in those 2 years. We didn't see any symptoms of crown rot. We did recover fusarium and PAM from some of those plants, but like I say, they were not sick. And so we weren't able to tell whether there was any effect of those treatments, those foliar fungicides or those in-furrow treatments on crown rot or yield. So that was unfortunately very disappointing. So yeah, thinking about those field trials and thinking about especially the in-furrow treatments and then also the seed treatments, and thinking about PAM and how PAM might be playing a role in crown rot, and assuming that that infection is happening early on in the growing season, we actually did some fungicide sensitivity testing with PAM. And so basically what we did is we had some isolates of PAM that we grew in the presence of the fungicides. And the fungicides that we used were fungicides that are commonly found in seed treatments or as an in-furrow treatment, right? And so you can see the list of the fungicides there that we had, the Fungicide Resistant Action Committee group that they belong to. So 1, 3, 7, 11, and 12, we have is that 6 groups of fungicides that we use on seed, on corn. And then you can see whether those fungicides, whether that PAM was sensitive to that fungicide or not. And yes, PAM was sensitive to all of those fungicides, apart from azoxystrobin, which I'll come back to. So that was good news that PAM is sensitive to the fungicides. So azoxystrobin is part of the QOI group of fungicides, or the strobilurin group of fungicides. You've probably heard a lot about them because fungicide resistance to that group of fungicides has been reported in numerous field crops. For example, frog eye leaf spot on soybeans, right, is resistant to the strobilurin group of fungicides. And what we found was that half of our PAM isolates were sensitive to azoxystrobin, and the other half were resistant to azoxystrobin. They grew perfectly well in the presence of azoxystrobin. And so we're following up on that research. There is a very common mutation that can occur in fungi that allows them to become resistant on strobilurins. And so we're looking to see whether that mutation is occurring in some of our isolates and not in others. And then if if we are able to see that, it'll also be very easy. It'll also be very interesting to see where the resistant isolates have come from and where the susceptible isolates have come from. And does that have anything to do with geographic location or history, cropping history, etc.? So, Tamra, I'm going to pass this over to you because you've been doing some really interesting work in the field as well, especially on early crown rot.
Hmm. Well, thank you, Allison. And, building on some of your earlier comments, we really do value the feedback and observations that people have described to us, our farmer colleagues, our crop consultants, everyone out in the field who has dealt with crown rot. And they've made a lot of observations. For example, often seeing more crown rot when we've had a cold spring, for instance, and we've tried to duplicate some of those scenarios and tested plants at different cold treatments at different stages, at germination and afterwards, and we've continued to fail to see crown rot symptom development, even in native soil where it was collected from fields with a history of disease. So we're trying a lot of things and beginning to look closer at the beginning of the season and what might be going wrong. In some of the other work that my students have done, they were looking in more of an exploratory way at symptoms in young plants. And so that's what we're showing in the photos here on this slide. And so they were able to find symptomatic plants that might look like they just had a little bit of seedling disease, a little bit of stunted chlorotic leaves that just didn't look healthy. They weren't thriving in the field. And when they dug those plants up and split them, they actually did find some crown rot symptoms in V4 and V6 plants. And so that in that third photo shows you what crown rot looks like in a seedling plant. And that was a bit of a surprise for us, but we think this could be important because this might be telling us that symptoms or at least infection might be occurring much earlier than what people might expect, since we don't normally see above ground symptoms until very late in the season. And so, looking at what they learned there, they wanted to look a little bit deeper into crown rot development or other symptoms and fungi in those plants early in the season. So in 2023, from those same plants and some of the work that they did, they extracted fungi from those symptomatic plants and ones that nearby or neighboring plants that did not have symptoms. And so the differences here in this graph is basically showing you the frequency that they were isolating each of these 6 different fusarium species from plants that either didn't have symptoms and appeared healthy, those are the orange colored bars, or the ones that had symptoms where, like I said, they were stunted or discolored in some way, not thriving. And those are the blue bars in the graph. So you have 6 different fusarium species that they isolated, as well as a little bit of Pythium and Calototricum. And you probably recognize those names as well. Pythium causing a lot of root rot and Calototricum causing anthracnose. And so it wasn't a surprise to find any of these there, but consistent with what we had seen in the earlier experiments, they saw more Fusarium graminearum from the symptomatic plants that were collected at that V4 to V5 or 6 time period than anything else. And so that still makes us look back again at that group of Fusarium species. And so, as an example, too, the picture of the 6 red Petri dishes, that's actually what Fusarium graminearum looks like. And I don't think we mentioned it earlier, but if that name rings a bell with you, for instance, this is also a common pathogen that can infect and cause disease of wheat. We call that Fusarium head blight or scab, although it's unclear if it has a role in this disease or not. And so the next step was for our students in 2024 to go back to some of the fields where we had a severe history of crown rot. And some of the worst crown rot here in Nebraska, anyway, has been observed in the western part of the state. And so, working with our Bayer crop science reps and three different farmers in two counties out in western Nebraska, Keith County, where there were two fields that they observed over the season, and one in Lincoln County. They watched two different hybrids, one that was identified as resistant to crown rot and a second one identified as susceptible, because those two hybrids were repeated in strips across the fields in all three of these locations. And so what they did is early in the season at V3 or V4, they identified pairs of plants, plants that were symptomatic plus a neighboring plant that appeared healthy, and they marked several hundred of those plants. They followed their progression through the end of the season, returning to record lots of data, symptoms, height, and a lot of other uh, parameters. And also collecting plants at five different stages from the V3, V4, all the way up to the end of the season at R5 when we normally see crown rot. And this was in an effort to see, can we narrow down when symptom development is and even infection? And again, who's at the party causing the problems early on? And at the end of the season, they split all of these crowns open, looking for evidence of stalk, crown, and root rot, and then analyzed that statistically. We think these results are kind of exciting because it gives us a little bit of hope that we might be looking in the right direction for some of these answers. And so there were differences that they observed in the resistant hybrid and the susceptible hybrid. That gives me hope that we know hybrids are behaving differently. And so that might help us tease apart what's really going on with disease development and infection. In these three locations, you see that labeled at the bottom of the bar graph, Lincoln County 1, Keith County 1, and Keith County 2 from left to right. These two graphs are showing the proportion of plants that had crown rot symptoms at the end of the season at R5. And what we found is that sometimes, in at least the resistant hybrid, in two of the locations, those early symptoms at V3, V4 were pretty good predictors of which plants did develop crown rot later in the season. And so on the left-hand side of the graph, on the upper half of the graph in that resistant hybrid, you can see the ones that had symptoms at V3, V4 were far and away the higher incidence of crown rot at the end compared to the ones that did not have symptoms. That was in Lincoln County. Keith County was similar, but not nearly as drastically different. Unfortunately, though, it was very different in the third location, the second one in Keith County. And one thing I want to point out is that in that Keith County location number 2, the incidence of crown rot at the end of this season was extremely high, no matter which hybrid you were looking at. 80% of those plants, no matter whether they were resistant or susceptible or had early season symptoms or didn't, developed crown rot. And so this is one of the most extreme cases where we've seen. And so I don't think we've provided a lot of answers here, but again, I think we're looking in the right direction, and it might help us tease apart some of these other questions that we still have rolling around in our mind about who's at this party. We also want to gratefully acknowledge, we could not do this work, both Allison's lab and ours, without the generous support from our sponsors. Bayer Crop Science has provided a lot of the support that has helped us do work in both of our states and provided samples and a lot of counseling. We've worked with them for a few years now, trying to talk through this. I don't think we're done. There's still more work I think that needs to be done to try to answer these questions, but we really appreciate their help and support doing so, as well as the Nebraska Corn Board, who's also been supportive of work here in Nebraska. Thank you.